Yeah, hi winners. I hope you are ready for the session. So today we are going to take the topic. So basically, I want to discuss more about DNA today. Okay, and I think it's better today if you're clear with my voice and with my slides. I'll be proceeding. Yes, I hope you have seen the basics of the DNA and RNA, which were posted as a PDF on your group. And I hope you have completed that. But I'll try my best to make things as simple as possible. So if you're ready, please drop me done in the chat box so that I can proceed with the class. Yes, okay, perfect, right. so now we are going to deal about the DNA and RNA, the genetic material, mostly that moves around the genetic material. And I feel this is an important thing which is missing in many basic MCQ books for a dental student. Okay, so carbohydrates, proteins, lipids, yeah, okay, acceptable. Vitamins, okay, acceptable. But this part, particularly the DNA, RNA, which is not at all covered properly in any of the MCQ books what we have. So that is the reason why I was stressing you to learn more as much as you can. This is nothing new, very new to you, because we have done our best to learn this in our 11th and 12th standard, but we are adding few more things and basically MCQ point of view. So what is this? So this is called as central dogma of the life. What is it? Basically, it's just the DNA. DNA is going to convert into RNA. The process of conversion of DNA to RNA is called as transcription, and the process of converting RNA to the protein structure is called as translation. So this is just a transcription and a translation. And DNA can generate DNA by a process called as replication. And I hope you know when we talk about the HIV. Okay, so that is where the reverse transcriptase means it is reverse to the process of transcription. What is actual transcription? DNA to RNA. What is reverse transcription? RNA to DNA. And similarly, like DNA, RNA will also undergoes a process called as replication. So this entire sequence of conversion of DNA to RNA, RNA to the protein is called as central dogma of the life. So this is the first thing that you have to make it. And when we are talking more about DNA, RNA, always we give more importance to the DNA. The reason why we give more importance to the DNA is more stable. So whenever the people who are more stable, we give more importance to the people who are unstable or less stable. Right? So that's the reason why we give more importance to the DNA. Okay, even the proceedings, even today's class, we give more importance to that of DNA. So why DNA is more stable? So this is the structure of DNA, okay, and this is the structure of RNA, deoxy. Okay, deoxy is an extra thing. for DNA. So what happens? Since this DNA, okay, so what happens is if you see this second place, okay, you can see the second place, which is different both for the DNA and RNA. In DNA you have H, okay, in DNA you have H at the second place, where in the RNA you have OH minus. So whenever you have OH minus groups, the atom or the molecule will be more prone for hydrolysis. So whereas this OH minus is absent in the case of your DNA, where H is present, so it is less prone for hydrolysis. That's the reason why DNA is more stable when compared with that of RNA. Clear? So DNA is more stable when compared to that of RNA. So this is the reason why we give more importance to DNA. DNA is more stable when compared with that of RNA. So I'm done with this particular slide or with this particular concept, and I want a response from your side, because compared to yesterday's session was roughly 20 minutes. This session will be a lengthy session, and I want you guys to respond at every level. Every 5 minutes or 10 minutes I want your response, because I don't know what is happening on other side, because if I keep a device near to me, it's going to create a sound. Okay, so that's the reason why I'm keeping all my devices aside and I'm just sitting in front of my desktop. So that's the reason. So I want your responses. So if your responses are fine, then I can go ahead. So I want your responses from in the chat box that yes, done. I want done from your side so that I can proceed further. Okay, I'm very good, very good. Compared to yesterday's live strength, today we have more than 120 or 130. It's crossing 130. It's really wonderful. Okay, yes, perfect, perfect. I'm going ahead. So the next important step is, yeah, we are more familiar nowadays with RRR. Okay, the Rajamouli's movie RRR. Similarly, our DNA is also having some RRR. So what are the three R's that are a part of DNA? Is DNA has the power of replication, DNA has the power of recombination, DNA has the power of repair. Okay, so all these three are the properties of DNA. They are called as RRR. Similarly to this, you have one more RRR, okay, which is a part of your microbiology, which we are going to discuss in the next month. But if you want, you can make a note of that RRR. Okay, that is leptospirosis. Okay, there is something called as leptospirosis, which has three R's. One is rainfall, R for rainfall. Okay, the second one is R for rat. Okay, it is caused due to rat, and the third R is rice field. It is commonly associated with the rice field. It is rainfall, rice field, and the rats. That is the other RR, that is leptospirosis. So next is, what are the individual components of DNA and RNA? Well familiar, you're well familiar with this. You can see RNA contains adenine, guanine, cytosine, and uracil, whereas DNA contains the same adenine, guanine, cytosine, and thymine. So that is the basic difference, structural difference, the content difference. The base pairs that are differ in RNA and DNA are uracil and thymine. In RNA, you will have uracil, whereas in the DNA, you will have thymine. And they can ask you a question like, which of the following base pairs are common for both RNA and DNA? The answer is adenine, guanine, cytosine. Okay, which are differ? The differ one is uracil in RNA and thymine in DNA. So next is the most familiar thing that you encounter, that is your DNA, the double helical structure of DNA. I hope you are familiar with all the things. And this concept of double helical structure of DNA was proposed by Watson and Crick in the year 1953. Please underline, Nobel Prize in the year 1962 is again a most commonly asked question. And why this is more important? Because this is one of the milestone in the modern biology. So that's the reason why they asked the question like, Nobel Prize was given in the year 1962. And yes, of course, we have discussed about the different types of DNAs, that is aDNA, bDNA, and zDNA in your PDF, very, very clearly given. But what Watson and Crick has given to us is the model of bDNA. They talk more about the models of bDNA, and most of your questions in the examination also moves around bDNA. If they don't mention, the question is about bDNA. If they clearly mention aDNA or zDNA, then we can think of, okay, this is question related to aDNA or zDNA. But if they don't mention, the question is most probably related to the bDNA. So DNA is a right-handed double helix. It contains of two polydeoxyribonucleotide chains. You have two chains twisted around one another. It's just like twisted around one another on a common axis. And I hope this is the most common familiar diagram of the double helical structure of DNA. And if you compare both the strands, okay, if you imagine that these are the two strands, okay, one strand and compare other strand. The two strands are opposite to each other, okay. The two strands are opposite to each other. They're opposite. The opposite means in the sense like they are anti-parallel to each other, such that one strand runs from 5 to 3 and other strand runs from 3 to 5. Okay. The next goes clear. Everything is clear and underlined. You can see two strands. The black color strand, that is 5 to 3, whereas the pink color strand, that is 3 to 5, they're running in opposite direction, that is anti-parallel. And the next one is the width of the double helix. Okay, if you see the width of double helix, see this width. The width of double helix is 2 nanometers or 20 angstrom units, and each turn or each helix is 34 angstroms. You can see this is each helix, which is 3.4 nanometers or 34 angstroms, and this each pitch or helix, it contains 10 pairs of nucleotides. Means each nucleotide, if they ask the question, if it is each nucleotide 3.4 nanometers by 10, then you will get the distance between each pair of nucleotide. If they ask 10 pairs of nucleotide, then the answer will be 3.4 nanometers. I hope you're clear until now. Okay, so you can see that these bonds. Okay, so you have a bond between A and T, G and C, T and A, G and C. All these bonds are hydrogen bonds. Okay, so the two strands hold together by hydrogen bonds. And if you see the side bonds, okay, you have side bonds also similarly, not bonds between the two base pairs, but you'll have side bonds. These side bonds are called as phosphide diester bonds. Okay, so the central bonds which holds the two strings together is hydrogen bonds, and the side bonds which holds the single string are phosphide diester bonds. I hope you're done with this also. I need a response from your side so that I will move ahead. Okay, so please do drop me a thumbs up or done or some clear word, some hint, so that I can proceed further. Yes, very good. We have crossed 150 plus today for the live session. Sounds good. Sounds very happy. So I want a thumbs up sign from you so that I will move ahead. Yes, perfect. Right. Okay. So next goes is the two polynucleotide chains are not identical, but they are complementary to each other. Okay. Yes, of course, it's very simple, but I'll make you understand, like we have some problem solving area also where you will get to know like what is complementary for this. Okay. So they're just complementary, means they can bond to the complementary. They cannot bond identically because A cannot bond a bond with A. Okay, A should have a complementary to bond. Okay, so it is not identical, it is complementary. The two strands are held together by hydrogen bonds formed by a complementary base pairs. That is, A can form a bond with T, and it has two hydrogen bonds, while G can form bond between C, which has 3 hydrogen bonds. So they can ask you, which of the following complementary base pair is more stronger? So the answer is C and G. because C and G has 3 hydrogen bonds. So if you want to break the bond between the C and G, you require more energy when compared with a bond between A and T, which is just 2 hydrogen bonds. Okay, so clearly given that Cg is stronger by about 50% than A and T. Okay, yes, perfect. Next one. The hydrogen bonds are formed between the purines and pyrimidines only. So there is some rule, we are going to discuss that. That is Chargaff's rule. What does Chargaff rule says? Everything we'll be discussing in further slides. And the genetic information resides on one of the 2 strands known as template strand. Okay, so that is all about Chargaff rule. Is the number of A, you have number of A's are equal to T's on other side, and number of G's are equal to C's on other side. So this is called Chargaff's rule. We have few problem solving area on Chargaff's rule, and we are going to discuss that in further slides. So we have already discussed that R is related to that of the properties of DNA, and the first important property of DNA is the replication. It has the power to replicate. For example, if you take the parent strand into consideration, this is the parent strand, where the parent strand is going to give rise to 2 daughter strands, in which if you take only the 1 daughter strand, half of the daughter strand is newly generated and half is the parent. Means you will have 50% of a parent in your daughter strand. So that is the best property of DNA replication. If you compare your daughter strand to that of the parent strand, the daughter strand will have 50% of the material same as that of the parental strand. So that's what is about. And that is the reason this form of replication is called as semi-conservative. Semi-conservative. Why it is semi-conservative means half of the original DNA is conserved in the daughter strand. Half of the original DNA of a parent, half of the original DNA of the parent is conserved in the daughter strand. So this type of replication of DNA is called as semi-conservative type of replication. Okay, and I hope you're clear until this level. So I want a response from your side, because from now everything has to be explained by writing something, because this DNA replication in the prokaryotics and in the eukaryotics is of course the most trickiest thing. So I made some notes to explain it as simple as possible to you. So I just want a response from your side so that I will proceed further. I hope you're clear with my voice and everything. Yes, I want just to have a quick recap what was done until now, so that we'll proceed. Okay, so what is central dogma? You're very clear. DNA is going to convert into RNA. RNA is going to convert into protein. DNA conversion to RNA is called as transcription, whereas RNA converting into protein is called as translation. We give more importance to DNA because DNA is more stable, and you know the reason, because second carbon atom you have H in the case of DNA, whereas OH in the case of RNA. So OHs are less stable, so RNA is less stable when compared with the DNA. There are 3 RRs which are related to that of the DNA, that is called as replication, recombination and repair. Similarly, in microbiology, leptospirosis are also having 3 Rs, that is rat, rice field and rainfall. So coming to that of the base pairs, in the case of your RNA, they are adenine, guanine, cytosine, uracil. In the base pairs of DNA are adenine, guanine, cytosine and thymine. Double helical structure of DNA was given by Watson and Crick in the year 1962. They were awarded with the Nobel Prize. And yes, it is anti-parallel, 5 to 3 in one direction, 3 to 5 in other direction. The width is 20 angstroms, and each turn or helix is 34 angstroms, which contains 10 base pairs. All about the hydrogen bonds. A and T has 2 bonds, and C and G has 3 bonds. But the bonding is primarily between the purines and pyrimidines. Some note about Chargaff's rule, and the basics about the DNA replication. Clear? Done? Yes. Hope you're able to see my blackboard. Is it visible for you? Hello? Conquers are able to see this blackboard? Yes, perfect. So we'll be starting. So I was telling that, so the topic here is the DNA replication. So the DNA replication is different in prokaryotics and eukaryotics. Okay. So most of the steps are, they look similar in a broad view, but basically if you go to a microscopic level, it differ in prokaryotics and the eukaryotics. Okay. So I'll try my best to make it as simple as possible in MCQ point of view. So until now we have discussed that the type of replication in DNA that is particularly prokaryotics is semi-conservative. Semi-conservative in the sense that when you compare with the daughter nucleus to that of the parent, you have 50%. So that's the reason why it is called as semi-conservative. And if you take that prokaryotics into consideration, so most of your prokaryotics, they have they have a cellular DNA like this. Okay, they have a cellular DNA like this, your prokaryotics, and for the initiation, for the DNA replication process, the initiation starts at some point. Okay, starts at some point. That some point is always a single point. So initiation is at a single point. Whereas in the case of eukaryotics, the initiation will be at multiple points. The first point to be added, the initiation point in a prokaryotic is always a single, whereas the initiation in a eukaryotic is always a multiple process. I hope you're clear about this. So there's something called as DNA. So DNA is going to come and it's going to start the process by attaching to that of the single point. For example, if you consider this as a single point where your replication is going to start in the prokaryotics, this DNA A molecule, okay, which is a molecule which is going to activate the process, is going to come and attach to that of the single point. Okay, so what happens is it's going to get activated. Your process going to get activated. Once this process is getting activated, because basically your stands in the most easiest way, because it's a double stranded circular DNA. So the strands will be like this, right? And it goes on and it comes and it's going to bond like this. This is a double stranded DNA, and we are taking this particular part and I'm expanding this. This is like this, right? So this is a point where it is being activated, and at the activating site we got our friend, that is DNA A. It comes and activates the process. Clear? So it comes and activates the process. What's going to happen in the next level? So this activation converts the normal DNA strand some bulge looking like some bubble like, right? So this is called as replication bubble. This is called as replication bubble, which is getting activated at the active site by DNA, right? So what happens if you consider only this part, or if you consider only this part, how it looks? It's look like this, right? So how it's looking? It is looking like a replication fork. It is called as RF. You have two RFs, one here and the second one here. Yeah. So this is called as replication fork. You're clear? You're clear with this? The bubble get activated, your replication fork and everything. So if I consider only one replication fork, okay, here it is 5 to 3 and it is 3 to 5, right? So because we have already discussed that the DNA is anti-parallel, one stand from 5 to 3 and other stand from 3 to 5. Clear? Done? Yes. The next one is the DNA helicases. So this is one of the important point to be added. This DNA helicase is also called as zip opener, because it opens the double stranded DNA. It is get activated here. So once it get activated, it is going to open the entire strand. How it opens? This is the entire double stranded circular DNA, right? So once it get activated, this is going to open entire strand and make the entire strand into a single stranded. So once it gets single stranded, it's going to give rise to the daughter cells. So the DNA helicases are also called as chip openers. So basically this DNA helicases are the enzymes bond both to that of the strands at the replication fork, and these helicases moves along the double helix DNA separating the two strands, which I have already discussed in the diagram, and they function as a zip openers. Of course, this DNA helicase strands depends upon ATP for the energy supply. So once this DNA helicase strand activates and entire zip is opened, you have two strands, right? This is RF, and this is a position where it's get activated, this DNA helicases, they get activated, and this helps in forming one strand, that is 5-3-, and other one is 3-5-. So one strand is called as leading strand, also called as continuous strand, also called as forward strand, where the synthesis of DNA is continuous, means no breaks, continuous. The second strand is called as lagging strand. It is called as discontinuous strand. It is called as retrograde because it starts from 3' to 5', and the production of DNA here is discontinuous. There it is continuous. You can see a continuous strand. Here it is discontinuous. So the small fragments, okay, so the small fragments, these are all small fragments. The small fragments of discontinuous synthesis DNA is called as OKAZIKI, Okazakis DNA. Okay, the small strands of discontinuous DNA which is present on the lagging strand, is the small discontinuous DNA. Because you want your daughter cell to be perfect, make sure that this discontinuous DNA should be converted into a proper, well-organized DNA. So one side we got this continuous strand, on other side we got a discontinuous strand. I hope you're clear until now. Okay, so in examination point of view, okay, we are done with this. In examination point of view, the most important things to be added is DNA polymerases. For now, we are learning about the prokaryotic DNA replication. So in this prokaryotic DNA replication, if you talk about the DNA polymerases, you have 3 types of DNA polymerases, out of which the third one is more important. So what third one do is, the first function of third one is, it helps in catalytic function of 5'3' cycle. Means this helps in catalysis. The 3 helps in the catalysis of 5' to 3'. That is the first primary function. And the second function is proofreading. So apart from the continuing the process, it observes like if there is any abnormality in this particular sequence or in this particular cycle. So if there is any abnormality, it's going to do some modifications. If there is any abnormality that has to be removed, it's going to remove it. So these are the 2 main functions that are done by the DNA polymerase 3. What it's done, it helps in the catalytic function of 5' to 3'. Apart from this, it helps in the proofreading. So we are left over with 1 and 2. So what 1 is going to do? So until now the process which we have learned is replication, right? So it has, the replication initially has initiation, elongation and termination, 3 steps. So the initiation is done. Now your RF area is going to get activated by helicases, and this extends to a process called as elongation. This elongation is continued by a primer. Okay, so the primer is an RNA primer. The RNA primer is going to continue the process of elongation, right? So the RNA primer is going to continue a process of elongation, and at the end there is someone who is going to come and terminate this process. The enzymes which are going to come and terminate this process are called as DNA tropoisomerases. Okay, so the 3 steps. One is activation. Activation is done by DNA A at the activation site. Only one site is there for the prokaryotics. Okay, and the process get initiated by helicases. Helicases are going to continue, and this continuation of elongation is done by RNA primer. So at the end, the RNA primer which is continuing this process has to be replaced by something. Once it is replaced, the process of termination should come into existence. The termination is done by tropoisomerases. So 3 steps. What are the main activities? You have to make it out. So now we are at the process of elongation, where the primer is going to come and primer is going to help you, and the primer is going to continue the process of elongation. So the main function of type 1 DNA polymerase is it's going to remove the primer and its position, and it's going to take the position. So the primer is going to continue the process of elongation. So once the elongation process is done, the primer has to be removed. The removing of the primer is done by 1. So DNA polymerase 1 is going to remove the RNA primer and takes its position. Apart from this, DNA polymerase 1 also helps in the catalysis of 5' to 3' direction fragment of DNA that replaces the RNA primer. So apart from doing the replacement, it's going to continue the process of DNA replication. Okay, so these are the two functions which are done by the polymerase 1. So this is an important area for MCQ point of view. 3, which is at most important because it helps in the catalytic function of 5'3 and proofreading. 1 is important because it's going to remove the RNA primer, which is the responsible for the elongation of this cycle. Okay, so at the end, what has to be happened is, so we are done with the initiation, we are done with the elongation, and we are about to terminate the things. So to form a bond, there is another enzyme which is required, that is DNA lyase. What DNA lyase is going to do is, it's going to cause the catalysis formation of phosphate diester linkages between the DNA, which is synthesized by that of DNA polymerase 3. DNA polymerase 3 has synthesized, and you need to attach it and form a bonds. You need to form a bonds, and that formation of bonds between them is done by DNA ligase. Okay, and last but not the least, we have missed 2. 2 helps in the repair of DNA. So at the end, what is going to happen? So now what this is your strand DNA on one side, easy to see, but on other side it is circular. It's coiled. Overall it's coiled. So if you take this part into consideration, so it is something like this, where the process of initiation has started. So the space between them has increased. So once, if you tie a knot, for example, if you're tying a knot at one area, and if you're keeping pressure at other area, if you're keeping pressure like this, another area, this gap is going to hit, go, and at this area, there will be formation of supercoils. Because these supercoils are formed because the main reason is DNA is a double helical structure. And if the replication proceeds from one end, and if it proceeds from this end, and it keeps on going, the supercoils are formed at the other end. This is the other end where the supercoils are formed. And the easy way to make you understand is, the formation of the supercoils can be best understood by comparing the DNA helix structure with the twisted ropes, one over the other. Hold the ropes at the tie ends in a fixed position and let your friend pull the other end. So what is going to happen? At the other end, you will have some coils which are going to increase the stress. So such coils are called as supercoils. And to break this supercoil and to end this process, we need DNA tropoisomerase. So DNA tropoisomerases are required to end this process. There are 2 types, that is type 1 and the second one is type 2. Type 1 tropoisomerases, they cut single-stranded DNAs. And type 2 tropoisomerases, they cut both the strands of DNA to overcome the problem of supercoiling. And these tropoisomerases, what I have discussed, are the areas where the cancer drugs are going to act in your pharmacology. So we have learned about anti-cancer drugs or anti-cancer therapy. So few anti-cancer drugs, the target area is this topoisomerases, where they are going to act on this topoisomerases is the target area which helps in the treatment of cancer. So this is the overall replication of DNA process in the prokaryotics. So I'm just going to have a quick decent recap so that you will have a better comment. So prokaryotic DNA replication is called as semi-conservative because the daughter DNA is going to have 50% of the content of the parent. Done. Next one. Next one is, it is a circular DNA, okay, circular DNA, and it has only one activation site where DNA A is going to come and activate. So once DNA A is going to come and activate, this is going to activate and this is going to form a bubble. And if you take one cross section of bubble, you will have your RF. RF is nothing but replication 4. And if you take that RF, the RF alone into consideration, your RF will have at the RF site, your DNA helicases are going to come and act. They are called something called as zip openers. They are going to open the entire strands of DNA like a zip. They are open. So you have 5 strand to 3 strand, and the 3 strand to 5 strand that is secreted is called as DNA B. So you'll have your DNA B secretion or the synthesis. So one strand is called as 5' to 3', and other strand is called as 3' to 5'. 5' to 3' strand is called as leading, continuous, forward, whereas 3' to 5' is called as lagging, discontinuous, retrograde. Whereas 5' to T' you will have a continuous one, whereas here you will have a discontinuous one. And this discontinuous DNA is called as Okazaki. Next important area to be focused is DNA polymerases 1, 2, and 3. 3 is more important because it helps in the catalysis of 5' to 3'. Apart from that, it helps in proofreading also. If there is any abnormality, any defect, it's going to remove or it's going to correct. But you have one more person here, that is RNA primer. Your RNA primer is there here, which is going to help in the synthesis, which is going to help in polymerization, which is going to help in the process of your RNA primer, which is going to help in the process of elongation. It's going to help in the process of elongation, is going to be removed by the type 1 polymerase. And finally, we got the outcome, but we need someone to bond them. Okay, the bonding process is done by DNA ligase. And at the end, you'll have so many supercoils because one end you're working, other end you're leaving. So once you start working on other end, as it is a double helical structure, you will have more stress on other end. The stress on other end is anatomically seen as supercoils. So to break these supercoils or to remove supercoils, you need to have DNA tropoisomerases. You have two types. One is type 1 and type 2. Type 1 helps in breaking single strand. Type 2 helps in breaking both the strands. So once the tropoisomerases activate, this bond is going to break and the stress is going to relieve. So this is a rough idea of DNA in prokaryotes. I hope you're clear with this. So once you're clear with this, please do drop me a text in the chat box so that I can proceed further. Okay, done. we'll go with the second synthesis. Until now we have discussed about the DNA replication in the case of prokaryotics. So now we are going to discuss about the DNA replication in the case of eukaryotics. So now the most oftenly I am just going to compare. Okay, so first of all, very important thing is in your prokaryotics, which is already done, the DNA replication takes place at a single site, whereas here it takes place at multiple sites, right? Whereas your prokaryotic, the DNA is a double-stranded DNA which is present in the cytoplasm, whereas here it's also double-stranded DNA. It's a circular double-stranded DNA which is present in the cytoplasm. Here the synthesis takes place in the nucleus, and you will have a huge amount of, very large amount of DNA, like roughly around like 1,000 times or 2,000 times DNA when compared with that of prokaryotics. And the synthesis takes place in the S phase. We have already learned about the we'll discuss about this in your embryology as well as in your radiology, that you have some cell cycles in which S phase is the phase where the replication of the DNA takes place. So the main problems here it is, your DNA is large in the prokaryotics, and your polymerases enzymes are very important. Your polymerase enzymes activity is slow. So DNA is large, you need to work on more DNA. Similarly, your polymerase activity is very slow. It goes very slow. So for this to occur, it's going to take so much of time. So that's the reason why the initiation starts at multiple stages. So that is the first difference you have to make a note. If you talk about the difference between the prokaryotic and eukaryotic DNA replication, multiple origin of replication is seen in the case of eukaryotics, whereas in prokaryotics there will be only single origin. The reason is the DNA is large and the polymerase activity is slow. So if you start at a single place, it goes very slow. So it's better to start at multiple places. Hope you remember your previous diagram with bubble. Similarly, you have multiple sites here. You have more sites here starting the process. These more number of sites are called as replicants. These are called as replicants, means they repeat. This is one replicon, this is one replicon, this is one replicon. And if you take each session of a replicon, you will have your replication fork. One acting on this side, one acting on opposite side. Clear? So there should be someone to activate this process. There in the prokaryotics, the person who has activated is DNA A. There it is DNA A. Here you have few terms you have to make a note. One is ORC. ORC is nothing but origin recognition complex. The word clearly reflects that it helps in recognition. The word clearly helps that it helps in recognition. The second one is RAF, that is replication activator factor. The word clearly helps that it helps in the activation. It helps in activation of the replication process. and the third one is RLF. So it is replication licensing factor. The word clearly helps that it helps in giving license, permission, the final permission. So one helps in recognition, and one helps in activation, and one helps in giving permission, that is license. So we have already discussed that there are multiple origins of replication. Means there is some area where the replication process is going to start, and all these three, ORC, okay, and the next one is RAF, and the next one is RLF. Okay, all these three together they form a complex that is called as Pre-Replication Complex, which is called as pre-RC. So all these three together they form a pre-replication complex. So you're ready with the replication complex, and this replication complex is going to go hit and it starts the process of your initiation, right? It starts the process of your initiation. So next comes, if you go back and have a look of your prokaryotic process, what we have learned in the just 10 minutes back, who is going to come and activate the next level of process, that is helicases. The helicases are going to come and activate the process. So similarly, we need someone like helicases in the prokaryotics, because now we are learning about eukaryotic. We need someone to come and help us. So those someone are MCMs. So what is this MCM? M for mini, C for chromosomes, and M for maintenance. So these are called as mini chromosome maintenance. You will have 6 of these. They are MCM2, MCM3, MCM4, MCM5, MCM6, and MCM7. 1, 2, 3, 4, 5, 6. So these are 6 MCMs which are going to come and help. These MCMs are analog to your helicases in your prokaryotics. So what these MCMs are going to do, these MCMs are going to unwind the DNA. So this is your RF. So these MCMs are going to come and helps in unwinding the DNA process, similarly like that of helicases in your prokaryotics. Clear? So next we need to go to the next level. So what is the next level? Next level we need to talk about the polymerases. There in the prokaryotics, what are the polymerases? 1, 2, 3. We have discussed already what are the functions that are done by each type. Similarly, in the eukaryotics, what we are discussing now, you have your polymerases, that is alpha, beta, gamma, you have your delta, and you have your epsilon. These are the main polymerases that are available in your eukaryotics, out of which we are not going to talk much about this. That is gamma. Gamma talks about the mitochondrial DNA. Easy to remember. M for men, G for girl. Okay, so men love girls. Easy to remember. So your gamma type of polymerase will talk about mitochondrial RNA, which we are not going to discuss anything about that right now, which is no way in the process of replication of RNA. So you are left over with alpha, beta, delta, and epsilon. And out of which beta helps in DNA repair. Now we are not talking about DNA repair also. We are talking about the replication of DNA. So out of this alpha, beta, gamma, epsilon and delta, gamma has no role because gamma is mainly concerned with mitochondrial DNA, and beta has no role because beta is mainly concerned with the DNA repair, and rest all, that is alpha 1 and epsilon and the delta are important. Okay. So basically you need to add a note about this, that is the delta. The delta is mainly concerned with lagging strand synthesis, whereas the epsilon is mainly concerned with leading strand synthesis. So please do make a note. The delta is mainly concerned with and the epsilon is mainly concerned with. So these are the two important things where they regularly ask questions. Okay, so what is that? Your DNA polymerase delta is mainly concerned with lagging, and your DNA polymerase epsilon is mainly concerned with leading strands. Clear? Yes, next one. The next important point to be added, both this delta and epsilon helps in forming a sliding clamp. So both this delta and epsilon together, they helps in forming a sliding clamp, which helps in fixing like a fixing. So this sliding clamp is something like it is a ring shaped clamp. Similar to your rubber dam clamp, to hold something, you should have a sliding clamp, and this sliding clamp is activated by this polymerase delta and epsilon, and this sliding clamp is called as PCNA. What is PCNA? Is proliferating cellular nuclear antigen. This is a ring-shaped sliding clamp which helps in the proliferation. So we are coming at the end phase. In the prokaryotes, as activation starts at one point, there will be formation of supercoils at other end. At that phase, what you have done, you have added topoisomerases to break that. But what happens in the eukaryotics? The initiation starts at multiple areas. So now here to end the process, the enzymes which came into existence are topoisomerases. Here to end the process, you will have endonucleases. These endonucleases are called as FEN1. So these are going to come and break everywhere to stop the process. So they start breaking. So once they start breaking, what is going to happen? You will have lots of gaps. The process is ended, but you will have lots of gaps. So these gaps they have to be filled. If you have gaps, they have to be filled. These gaps are filled by DNA lysis. So once this process is done, you need to terminate this. So the last step anywhere is called as termination. The termination will be done by telomerase or telomer. I have posted PDF. Okay, in the PDF you have 1 to 2 pages of notes related to the telomeres. So this is how the process of DNA replication ends in eukaryotics. So just have a quick recap. This is a bit tricky to understand, but I tried my best to make it as simple as possible, focusing mainly on the MCQ part. So first of all, coming to the eukaryotics, you will have an area where the process starts at multiple areas. Okay, so DNA is large, and your DNA is large, and your polymerase activity is slow in the eukaryotics, and that is the reason why we need to start the process at multiple sites. So this multiple site process, you can see in this diagram, it's a multiple site process, and each site is called as a replicon, and each site will have two RFs, one RF facing this side and other RF facing on other side. So before we go into the process, DNA A is the fellow which is going to activate the process in the case of your prokaryotics, but here we need a complex to activate. That complex contains ORC, RAF and RLF. All three together, they form a complex that is called as Pre-Replication Complex, which is going to bind at the active sites. The active sites are multiple. So they are helicases which are going to come and start the process like a zip openers in your prokaryotics, and here that process is done by MCM, that is called as mini chromosome maintenance. There are 6. The number starts from 2 to 3 to 4, 5, 6 and 7. So they start the process. So if you talk about the polymerases in the prokaryotics, there are 3 types, 1, 2, 3, whereas in the eukaryotics there are 5 types, out of which gamma is negligible because it is mainly concerned with the mitochondrial RNA, and beta is mainly concerned with the repair. So we need to talk more about alpha, delta and epsilon. So alpha and delta, they help in formation of leading strand and lagging strand, and sliding clamp is also found by them. The sliding clamp is called as PCNA, that is called as proliferating cellular nuclear antigen. And some basic differences, how in prokaryotics the process is ended by tropoisomerases. The suprahelical structures are break by tropoisomerases where it terminates, whereas here the endonucleases Fe and Fen is the one that's going to help in the activation. So once the Fen is activated, you will have multiple gaps, and these multiple gaps are filled by the DNA ligases, and the termination is done by tropoisom, that's telomerases and telomere. I hope you're clear with the concepts of replication of DNA. Okay, so I want to end the session at this particular point, and tomorrow we'll have one more session like talking about the different types of RNA and how the DNA, the complementary is secreted, and how the complementary is going to result in the formation of RNA, and how the RNA is going to result in the formation of a particular sequence of protein. So that is more interesting and more easy to understand when compared with this tricky subject of DNA replication, but it is very, very important. Missing, and I hope I have missed this even in my biochemistry. I hope my professor has not mentioned about this complicated process, or I'm not aware of this at that particular phase, but this is the first time we are taking a more to explain everything in detail because we have time. Okay, so signing off for now. Thank you all. I hope you need to work more on this area. So let's work today and tomorrow more on this particular area, and further you can go for the revision. And what I'm going to do from my side is I'm going to help you in covering few small topics, and most of these small topics which are left over in the biochemistry are a part in other subjects. For example, the biochemistry has hemoglobin structure also in it, plasma proteins also in it, structure of albumin, your globulins and your hemoglobin and everything are a part of biochemistry according to the syllabus, but these are going to be a part of your physiology also. When we talk about the blood, we talk about the structure of hemoglobin, we talk about the structure of globulins and everything. But anyway, I'll try my best to cover those things also, so that you will be more fit to the particular subject of biochemistry. Similarly, you have your BMRs. Again, they are part of your general medicine and your physiology also. But I'll try my best to cover that as a miscellaneous topics during your revision. But I want you guys to focus more on revision. Probably tomorrow we'll pack up the things related to the bio with few more concepts. I'll try to finish the session in the morning so that From tomorrow afternoon, you can start your revision. Revision is more important for the exam, which is on 5th. Main target is the exam on 5th. Okay. Signing off for now. Take care and all the best. Bye.